Good day. These are the sample questions for the 9th topic under aquaculture. Aquatic animal health. 1. This is an ultramicroscopic non-cellular genetic element which size ranges from 10 to 300 nanometers that requires a living cell in order to replicate. A. Pathogen. B. Virus. C. Bacteria. D. Microorganisms. The answer is B. Virus. Viruses are important pathogens of cultured fish and crustaceans. Mortalities associated with outbreaks of viral infections are usually high and have enormous economic impact. 2. The following are the major preventive measures to be practiced to avoid the contact of fish with pathogens, except A. Exclusion and elimination. B. Disinfection and prompt removal of infected fish. C. Identifying the predisposing factors that may lead to a disease state. D. Limit stocking density. The answer is D. Limit stocking density. A major preventive measure to be practiced to avoid the contact of fish with pathogen is by exclusion and elimination. Introduction of pathogen into the culture system could be prevented by disinfection and by prompt removal of infected fish. Another important consideration involves identifying the predisposing factors that may lead to a disease state. Once predisposing factors are identified, appropriate corrective measures should be initiated in the culture system. 3. Which is a sign of a stressful environment in fish? A. Erratic swimming behavior. B. Gasping of air. C. Inflammations. D. Skin lesions. The answer is B. Gasping of air. Erratic swimming behavior, inflammations, and skin lesions are signs of infections due to microbial pathogens, while gasping for air can be a sign of diseased fish due to microbes, but it could also be, or mainly also, a sign of stressful environment, specifically low DO level in the water. For which is not the causative agent of saprolegniosis? A. Saprolegnous species. B. Aphanomyces species. C. Myxobolus species. B. Aquila species. The answer is C. Myxobolus spp. Saprolegnous species, Aphanomyces species, and Aquila species are causative agent of saprolegniosis, while Myxobolus species is the causative agent of whirling disease in salmon and trout species. 5. The following can cause stress in fish, except A. Transporting fish in plastic basins. B. Scooping of fish. C. Stocking at low temperature. D. Stocking at high noon. The answer is C. Stocking at low temperature. All forms of handling can cause and contributes to handling stress. However, stocking at low temperature is advisable because it causes less stress to the fish than stocking at high temperature. 6. Which should be done if a fish lost appetite during the course of grow-out operation? A. Lowering of water temperature. B. Adding more feed. C. Adding of water. D. Lowering the stocking density. The answer is D. Lowering the stocking density. By lowering stocking density, it should be expected that the fish are reared in spacious environment which result to good water quality and healthy fish, which should not be a reason for them to lose appetite in feeding unless the other choices are experienced by the fish. 7. Which is the main gross signs of an epzootic ulcerative syndrome? A. Difficulty in breathing. B. Skin discoloration. C. Ulcerative lesions. D. Loss in appetite. The answer is C. Ulcerative lesions. From the name itself, main gross signs of an epzootic ulcerative syndrome is ulcerative lesions in the infected fish. 8. What causes pale or anemic color of gills? A. Poor nutrition. B. Parasitic infection in the skin. C. High nitrite toxicity. D. Low blood. The answer is C. High nitrite toxicity. High nitrite toxicity causes anemic or pale-colored gills. 9. Which does not belong in the following? A. High pH. B. Intensive fish farming. C. High organic load. D. High ammonia. The answer is B. Intensive fish farming, production of high wastes product resulting to increased or high ammonia level in the water, lower DO level, increased organic load, and high pH are results of intensive farming. 10. It is the process of adding fixatives to attach parasites in the glass slides that needs to be observed. A. Fixing. B. Staining. C. Killing parasite with the use of fixatives. D. Microscope observation. The answer is A. Fixing. Fixing, or fixation, is the process of attaching the parasites to be observed. Adding fixatives, heat fixation or chemical fixation, will not only attach the parasites to the slide, but will also kill them, preventing them to create movement while maintaining the integrity of their cellular component. 11. These comprise a group of microscopic, single-celled organisms living in nearly all the environments, particularly aquatic bodies. A. Pathogen. B. Virus. C. Bacteria. D. Microorganisms. The answer is C. Bacteria. Bacteria are found everywhere in the aquatic environment. Disease caused by bacteria may cause heavy mortality in both wild and cultured fish and crustaceans. 12. What type of culture media has special nutrients added to support fastidious bacteria? A. Enriched media. B. Selective media. C. Differential media. D. Synthetic media. The answer is A. Enriched media. There are 3 types of media based on function. Enriched media has special nutrients added to support fastidious bacteria. Selective media contain agents that inhibit or allow growth of certain bacteria, and differential media that contain growth agents that promote different phenotype of different organisms. On the other hand, synthetic media is a type of culture media based on chemical composition. 13 ways to isolate pure culture. 1. Serial dilution. 2. Spread plate method. 3. Streak plate method. 4. Colony isolation method. A. 1 and 2 only. B. 1, 2, and 3 only. C. 2 and 4 only. D. 1, 2, and 4 only. The answer is B. 1, 2, and 3 only. There are 6 ways to isolate pure culture: streak plate method, pour plate method, spread plate method, serial dilution, enrichment methods, and single-cell isolation. 14. Which type of colony margin has thread-like, spreading edges, has irregular indented edges, and has wavy edge with layered appearance respectively? A. Entire, erose, lobate. B. Filamentous, erose, curled. C. Curled, filamentous, undulate. D. Entire, lobate, undulate. The answer is B. Filamentous, erose, curled. There are 6 types of colony morphology based on margins or edges. Entire has smooth, even edges. Erose has irregular, indented edges. Lobate has highly indented, irregular edges. Undulate has regular, wavy indentations. Curled has wavy edges with layered appearances. And filamentous has thread-like, spreading edges. 15. Which of the following is incorrect based of the bacterial elevation morphology? F. Bacillus subtilis, unbonate. B. Enterobacter aerogenes, convex. C. Klebsiella pneumoniae, flat. D. Escherichia coli, slightly raised. The answer is C. Klebsiella pneumoniae, flat. The following are the basic features of Klebsiella pneumoniae culture. Colony shape and size, round and large margin, undulate elevation, umbonate color, white/translucent texture, muciid. 16. It is the study of animal tissues and how these body tissues are organized to constitute organ systems. A. Histology. B. Immunology. C. Parasitology. D. Cytology. The answer is A. Histology. Histology is the study of animal tissues and how these body tissues are organized to constitute organ systems. All animal tissues are composed of cells. Histology is just one of the methods to assess the health or diseased status of fish. 17. The following are some methods by which potential pathogens may be inactivated or removed in a culture facility. I. Inactivation of pathogens by means of exposure to ultraviolet radiation. 2. Removal of pathogens by means of microfiltration. 3. Infected fish must observe first before completely removing and destroying. 4. Elimination of chemical pollutants by means of carbon filtration, bio-filtration, and water dilution. A. 1 only. B. 2 and 3 only. C. 1, 2, and 4 only. D. 3 and 4 only. The answer is C. 1, 2, and 4 only. Some physical methods by which potential pathogens may be inactivated or removed in a culture facility. Inactivation of pathogens by means of exposure to ultraviolet radiation. Removal of pathogens by means of microfiltration. Elimination of chemical pollutants by means of carbon filtration, biofiltration, and water dilution. Exposure of tank or pond to heated water in sun drying can also eliminate some microbial flora, and infected fish must be removed quickly and destroyed. 18. Generally, there are more reported economically important bacterial pathogen in tropical countries than temperate countries because 1. There are more culture species in the tropical countries. 2. There are more cultured species in temperate countries. 3. There are more researchers working on bacterial pathogen in tropical countries. 4. There are less researchers working on bacterial pathogens in temperate countries. 5. There are more reported bacterial diseases in tropical countries due to higher temperature compared to the temperate countries. 6. There are more reported Bacterial diseases in temperate countries. Due to higher temperature compared to the tropical countries. A. 1 and 4b, 1 and 5c, 2 and 5d, and none of the above. E. All of the above. The answer is B. 1 and 5. Bacterial growth is usually faster at a higher temperature. Although there are cold-specific bacterial pathogens, there are more reported bacterial pathogens in the tropical region due to larger number of cultured species and higher water temperature that is very suitable for microbial growth, especially in intensive farming. Also, bulk of aquaculture operations of diverse species are usually present in tropical regions. 19. This is not recommended in aquaculture due to high development of antibiotics-resistant microbes in the aquatic environment. A. Use of antibiotics. B. Use of high-quality nutritional diet. C. Use of probiotics. D. Use of vaccine. The answer is A. Use of antibiotics. Use of antibiotics in the aquaculture operation is not recommended due to the risk of development of drug-resistant bacteria. 20. Salt bath treatment is applicable only to the following except A. White spot disease, ichthyopterias. B. Skin lesions. C. Ectoparasites. D. Endoparasites. E. External fungi. F. A and B. G. A and C. H. A and D. The answer is H. A and D. White spot disease, itch, cannot be treated with salt bath since they are present underneath the epithelial cells and not directly exposed to the water. Same also with the endoparasites, wherein they do not have exposure to the water, indicating that salt bath treatment will be useless.